Okay, good afternoon. My name is Dick Bolterman. I am the chair of today's session. I'm a board member of Informatics Europe, and I'm the person who is in charge of our Informatics Europe Distinguished Lecturer webinar series. Before we start with today's lecture, I'd like to spend a few minutes giving some background on Informatics Europe. And that'll, number one, provide some context on the organization and the series. And number two, provide a little bit of extra time for people who will be joining us to find their way to our Zoom connection. Who is Informatics Europe? We are a not-for-profit membership association. And our members are computer science institutions such as computer science departments, research institutes, faculties, and research labs. We also have a number of corporate members who have research interests in the area of computer science, either in core technology or in applications of computer science. The origins of our organization date back to a European Computer Science Summit, ECSS, which was held at ETH in Zurich in 2005. At that time in 2005, it was felt that the European computer science community really needed an organization that would bind together a number of national organizations and help lobby for the interests of the computer science research community in Brussels with the European Commission, and to help organize distributed events throughout Europe, maybe sharing expertise and connections between partner members. This organization was founded along the model of the Computer Research Association in the United States, and the CRA has been very successful as being a platform that has brought people together and also that has brought organizations together and built a common voice. Our goal is to represent the public and private research community of informatics and informatics researchers in Europe and in neighboring countries. Currently, we have members from over 160 institutions, departments, faculties, schools, institutes, and we cover 34 countries. You can see on the map here that our coverage in Europe, stretching from Iceland to Turkey and from Finland to Spain, or actually, I suppose Greece is more southern than Spain, is very broad. The coverage is complete. Belarus and the Ukraine have some other issues that they're working through right now. They are not members. There are some other countries where we're actively looking for membership. Certainly there's a strength in numbers, and what we'd like to do is get good national coverage so that we can combine European focus with also a national focus. What we do is create a common voice, and that voice is there to safeguard and shape quality research. We promote particular positions and acts of shared priority in the areas of education and social impact and research. I'll give you a list on the next slide. We foster knowledge transfer and collaboration, both with our member institutions and also with the staff members of those member institutions. Our activities are performed mainly by scientist volunteers. And these are people who commit their time and their effort to promoting the missions and goals of Informatics Europe. There are a couple of events coming up for those of you who are interested in Informatics Europe. One is this year's European Computer Science Summit that'll be held in Hamburg in Germany with the theme, the workforce for the digital transformation. We had a hybrid event in Madrid last year with a number of speakers on a broad range of topics dealing with sustainability. And that's available on the YouTube channel. We run this free Informatics Europe webinar series. And we also have an academic leadership development course. There's a summer cohort that's announcing, and the goal here is to help our staff members of our institution develop their leadership skills. You can follow us on Twitter. You can follow us on LinkedIn. You can watch us on YouTube. And for those of you with us today live, you can also be with us as we welcome this month's speaker. I will stop sharing my screen and take a moment to, let's see, reduce my size. And well, to welcome Professor Jaap Heringa. We've had a number of lectures in this series that operate on core technologies and on sort of traditional computer science themes, from software engineering through algorithmics, and last time we heard about distributed systems. This time we start a 3-webinar series that puts the person in the loop, and literally the person in the loop in the sense of bioinformatics. Next time we look at the human in terms of video conferencing and support for hybrid people, not just hybrid conferences in solutions. And in the third sort of lecture of this mini series, we look at human-centered AI. There'll be some announcements made on those lectures at the beginning of next week. So if you're interested, make room in your calendar. But right now, we'd like to make room for Jaap Heringa. Professor Heringa is chairman of the Department of Computer Science at the Vrije Universiteit in Amsterdam. He's had broad experience in a wide range of bioinformatics communities. I'm very pleased that he accepted our invitation to give some background on this area, which gives, I think, a very nice example of combining application area problems from a wide range of areas, data science techniques, the application of artificial intelligence, all with the goal of understanding how the biology of the systems in which we live actually work. So I think that Professor Heringa can even give a better presentation of what he's going to talk about than I can. So I'd like to shift him the screen. I'd like to welcome him. If you have questions, you can post some of them in the chat. But at the end of the presentation, if you have questions, please raise your hand, and I'll go through people in the order in which they appear on my screen. Okay, thank you for joining us, and Professor Heringa, welcome, and we look forward to your presentation.
Thank you very much, Dick, for this nice presentation. I hope you can all hear me. I'm now going to try and share my screen. I hope this works okay. So on the lecture, if there will be any questions, certainly if there are burning questions during the lecture, feel free to break in as well. I really don't mind. So Dick sort of suggested before that you know, this is the lecture, all there is to know about bioinformatics you were afraid to ask. I think at best this will be a short kaleidoscopic overview of what is there to entertain and what kind of problems we face in bioinformatics. But first of all, I'll introduce myself. My name is Jaap Heringa. And you know, Dick already said that I'm leading the Department of Computer Science at the Vrije Universiteit VU Amsterdam. But I can add, I'm stepping in the steps of Dick, because he was the earlier head of the department. So I must also say it's very nice to be present here via this webinar, but I would much rather have been with you face to face in a room, but these are still these times. So let me try and fill you in a little bit on bioinformatics and sketch some relationship with data science and for that matter, computer science. So if we take a very broad view of the history of science, maybe there are three phases that are sort of illustrated here. So the first phase, at least in the Western world, I think, in the Arabic world, mathematics was long there before we in the West started thinking about science, for example. But, early on, anatomy and architecture were the main fields of study. And this is illustrated here by a famous picture by Rembrandt van Rijn, the Dutch painter. This is the anatomical lesson of Dr. Tulp. And this was an illegal sort of event that took place regularly in those days, where people were, looking at a dead body that was dissected in order to learn about, in this case, the anatomy. So then something happened, and I'll illustrate this with an illustration or with an animation that I'm much proud of. There we go. Let's see if that works. If I can, if my computer helps, that would be very good. Yeah. Can I go back? Yes, now it works. So very good. So this is sort of the technology level you may expect today. So I hope that's not too taxing. So in 1726, something happened, according to some people, at least. That was that Isaac Newton was sitting under a tree and an apple fell on his head. And where normal mortals would say something to the effect as, ouch, allegedly Newton said, hey, F equals m times a. And by that event, dynamics were born as a field of study, and we know, and a lot of that has come since then. Of course, all the physics and things we know. Then maybe after, I should say, the Second World War, we have now to become more precise when it's about counting wars, I'm afraid. There was a new field of study by Norbert Wiener that you see also a picture there in black and white, a bit lower on this slide. Cybernetics was a field that was about studying control in machines and animals, and a lot of combinations of technology and the observable world. So there was a shift from anatomy to static shapes to dynamical shapes, and now thinking about the information content of systems. So in the wake of all these developments in this timeline, perhaps fields like, indeed, computer science, but I'm mostly talking today here about genomics, bioinformatics, and systems biology are a derivative of this field, of this development, I should say. So first of all, let's go to the origin of bioinformatics. A lot has been done. Norbert Wiener maybe was also a pioneering figure, but it was not called that so far. This is the city of Utrecht in the Netherlands. It has a nice canal structure, as you can see in the middle picture and in the top line, in the top row. So there the word was coined for the first time, and we know this thanks to a very obscure article that appeared in a student journal, an obscure student journal, I should say, and this paper was in Dutch. Why am I telling you this? Because around 1985, a few scientists in France started claiming that they had conceived the word bioinformatics for the first time. And, they might have done this independently, but here is really a piece of proof that they were just 15 years too late, that in 1970, Pauline Hogeweg coined the term for the first time. And I'm old enough to know that here on the bottom, there was really already a signpost on her door that mentioned Bioinformatics Group. Not only did she coin the term, she also gave sort of an apt definition that still holds today, you might say. It's about studying informatic or informational processes, for that matter, in biotic or in live systems. And that still holds very much true. So biological systems are information processing systems. If you would like to read about all of this history, there is a citation on the bottom there in PLOS Comp Bio. Okay, so we have then anatomy, dynamics, informatics as a natural development in science, but we see very much that modern life science, and bioinformatics is no exception, turn to become very much data sciences. And of course, they are becoming ever more interdisciplinary. So, apologies for putting bioinformatics as sort of the center of the world, but since I'm talking today, I'll take the freedom to do that. So bioinformatics is really a combined field, an interdisciplinary field, although these words are becoming moot by recently by this time. You have heard so many of these terms. But anyway, the play garden, you might say, what we try and understand is the molecular world. This is sort of the substrate we're talking about and trying to understand. And from that, all functionality happening in organisms or even larger communities of living systems. So what you need is molecules. So chemistry is naturally there. We talk all about physics, of course, so physical forces. So what we need to really try and understand that is and model all of the things we observe is mathematics and certainly also statistics to do data analytics. Computer science and informatics is inspiring, is, we use it, but sometimes we even give ideas back to the field from bioinformatics. And, maybe nowadays the most important application domain probably is medicine. And I'll get back to medicine and personalized medicine toward the end of this lecture. So more on the theme of what is really driving life sciences and bioinformatics, and really it's clever thinking, but most of all, it's just technology that has been developed, and certainly technology to do high throughput measurements of all kinds of things. So genomics, I'll talk about in a moment, various other high throughput measurements like sequencing, ChIP-seq, RNA-seq, that has all to deal with genomics. There's also the field about proteins, proteomics, about the metabolome, all small molecules, metabolomics, and a lot of technologies, X-ray, diffraction, nuclear magnetic resonance, mass spectrometry techniques are all used to measure particles in these areas. More recently, we've seen that great strides have been taken in imaging, optical measurement techniques, single cell measurements, for example, or even single molecule tracking, where it's like, and who's done it? A molecule, protein is tracked, it goes from one cell to the other and can just be followed. So this is all quite exciting. And importantly, a lot of new stuff is coming up. A lot of the measurement techniques are being in the making and will come also in the future. Things are going faster and all the time, I must say. So what, of course, is important to note, to make anything of these data and these measurements come to life, everything is enabled by IT, so that's totally fundamental for these technologies. I might say when you look at bioinformatics, you might also say it's a little bit of a nervous field. A lot of things to come together. It's changing all the time. The measurement techniques change all the time. There's new data. So any old idea you have is being refuted the next week when new data comes up. So we have new technologies, new formats, new standards, new hypes, and of course, hopefully at least very many new insights. If you, for example, compare this to another field of study, and I like Greek philosophy a lot, but maybe it's fair to say that the number of new data is a bit less and insights might develop, but this is all a slower pace. So, this might be a choice if you want to have a more relaxed life. Okay, a few fundamental questions here, and I'll show you some numbers to illustrate to you that bioinformatics is the science about large numbers. So here we see that most life forms start with a single cell, fertilized egg cell, a zygote, and then you get cell division. And in humans, for example, the single cell will in the end lead to roughly 42 trillion cells. That's the most recent estimate, I think. Before it was 50 to 100 trillion. So we're talking to 5 times 10 to the power 13. That's many cells. And what is more, you can see on the color diagram in the middle, is that there is differentiation from the single cell in roughly over 200 tissues in man, roughly 210 tissues that are being developed. There is not a central control tower sort of taking decisions as what cell will differentiate in what subcell and what form, but somehow in most cases, which is incredible, I think, you get people that are born in a reasonable shape and can even communicate together. So if you look at the complexity of the systems that we represent and how this comes about during the first 9 months, it's breathtaking, I guess. Okay, first question then, how do you get from a single cell through all of this complexity? Another point here, so to illustrate the enormous diversity in life, we have, of course, we've talked about that a lot, I guess, in the last 2 years or so, that viruses have been very much on the mind of people. But the virus is a sort of an incomplete life form. It's not life yet, it's part of life, and it needs a host to be turned into something that is about life. The first life forms are single cell organisms like bacteria and other unicellular organisms. Now, and of course, the more complicated or more complex organisms are multicellular. C. elegans is sort of a worm. I'm going to mention that in a few moments. And having 1,000 cells. But I think the blue whale and other animals can have hundreds of trillions of cells. So, and these animals are also live and kicking. So that's a big secret, how they do that. So as I said, it's the science of big numbers, 42 trillion cells for us, divided over 210 different tissues. In each of those cells, you may expect to have 42 million protein copies, and proteins are named the doers in the body. So lots of processes in the cell are carried out by proteins. Almost all cells also contain DNA and also many shorter functional RNA molecules. So it's quite a box, each cell, where a lot is happening. The complexity is, again, mesmerizing, and then you have to multiply that complexity roughly 42 trillion times for a human. And then next to the genetic machinery that I'm sketching here to you, there is also, for example, the gut microbiome, so all the bacteria that are in your gut, or maybe in your mouth, the oral microbiome. And that is, the gut microbiome is more complex and more divergent than our DNA. Your identity is more defined perhaps by your own microbiome, the things that are not you, the bacteria in your body, than by your own DNA. So this is all very interesting, and it's also known in the meantime that your health is for a large extent dependent on the health of your microbiome. And a lot of diseases, could be tracked early on by measuring the microbiome. So that's really very interesting. And Parkinson is an example in case where estimates are there that 30 years before the onset of Parkinson, there are patterns changing in the microbiome that might be picked up in not too long if we start studying it a bit more intensively. Okay, so basically we have this question. We have DNA in a cell, which is the blueprint of life. RNA is encoded by DNA. Proteins are encoded by DNA. So basically the stuff happening in the cell is encoded by this one template, the DNA molecule. And we know, for example, that musicality, you see a young violinist here, runs in families. So how can we go from this genotype, a DNA molecule that is basically in principle identical for all of the cells you have, to unless you would get cancer, then you get appreciable differences between cells in terms of the DNA. But anyway, how does a genotype lead to a phenotype? And this young lady being good at playing the violin. Okay, step back. In the olden day, bioinformatics started roughly in the 70s. It really came to fruition, you might say, in the 80s. So what happened early on? We were trying to find out about protein and nucleotide structure and function. So we started comparing stuff. Ever since Darwin, the comparative analysis has brought us very far in science. And so methodologies and methods were created to do protein alignments, to do sequence alignment, and all this to be able to relate pieces of life to one another and learn from that. Another important problem, I'll get back to that in a moment, I guess, is the protein folding problem. We have an enormous diversity of protein shapes. And so a protein is a sequence of beats on a string, you might say. There are 20 different amino acids, which are the building block of a protein sequence. And somehow a protein knows all the time how to fold up and how to assume a three-dimensional shape, structure. And from that comes that there is an active site somewhere on the surface of such a protein, such that it becomes a given enzyme, for example, an alcohol dehydrogenase that comes in place nicely when you've been out to a drinking spree the last night. So also what was an important question is, how do these proteins interact, and how do proteins interact with nucleotides? Because these are all very important activities and events happening every split second in the cell. So from all of these basic questions, a lot of methods were created. I already mentioned protein sequence analysis tools, protein structure prediction method, all types of levels of structure, secondary, tertiary, even quaternary structure, and also the interaction between proteins and so on. It's called docking to do something about that. There were also methods creating for that. Also, evolution has been a driving force in trying to make sense and understand the function of components of a system. So phylogeny, the evolutionary ancestry was studied, and it is really still a field of intense study. There are bookcases full of books on this, and also phylogenetic reconstruction by clustering methods. The neighbor joining technique is sort of an early famous one. in 1984 that was created. So a lot of people became kind of famous in the early years by creating a method. And that is, of course, a nice thing to hear for a computer scientist as well, that can happen. Okay, so we were making all kinds of nice methods, we think. We were analyzing data. It was not big data yet, but it was, appreciable shapes of sizes. We were trying to become important, but then something happened, a big bang in bioinformatics, and that was the Human Genome Project, taking place from the mid-90s, you might say, until the year 2000, when the world was united in this one project. So basically, at the time that this started, that people thought we should do this, this was sort of thought that it was utterly impossible to do anything like this. The computers were not there, the experimental setups were not there. So this was really conceived as a crazy idea. But then some diehards started pushing. And in fact, at the end, it was much faster and cheaper than anybody had thought, had been able to think of before. So that's quite a success. But then, we might also say, as a great claim, this was basically the first global, worldwide, collaborative and interdisciplinary life science project with big data exchange for the first time, perhaps via the internet. And so this sounds great. But then again, collaborative, and now we go a bit in history, might be taken with a grain of salt, because it was a bitter fight. What had happened? It turned out to be a nervous race between academic groups and industry cellular. I'll sketch this in a moment. So at stake were patenting issues. So the prospect of just somebody having all of the rights to knowledge and could impede the progress of all of biomedical science. So people felt at the time we were in dire straits. And the main character in all of this is Dr. Craig Venter. You might have heard of the name, who was in the NHS the NIH in the United States, but then became dissatisfied with how things were going there and founded in his own company, Cellera. Here we see, the man on the left-hand side, I guess you still recognize, even younger people, and you see an old-fashioned television screen there, we're talking the year 2000 here, but Craig Venter is in the lower right corner of this message. What was the threat here? Venter said, I will be the first to sequence the human genome, and I'll patent every gene that I'll find in that genome, and then you come to me and you pay dearly for anything you'd like to do. For example, solving or treating a given disease, if that would have a genetical base. So this didn't look very nice, and then people started just assuming that, how can it be that somebody could patent my gene or the gene of some person? That can't be. So of course there is legislation to prohibit this. So people started looking, and of course there were no laws. So what had happened? Craig Venter was already had founded this Cellera Genomics company. He had built the greatest computer cluster of his day, certainly in companies at that moment. Talking in the mid to late 90s, and he was happily sequencing, and much faster than the important places, the academic places in the world did it. So then an allied force was created, and the universities decided, and research institutions decided to join forces and try and go in a race and be just finish earlier than Craig Venter could do. So you might say a lot of new ideas and it came from that. The urgency was high, but of course it could have been a bit nicer in the world. It was really by competition that this all happened. So we take a big leap to 26 June the year 2000, when there was a great press conference when the two main players, so the Allied Forces, the Human Genome Consortium, and Craig Venter with his Cellera company said they were friends. It was a photo finish. They all did it together, and there was a nice press conference on the 26th of June. Here you see a couple of pictures. You see Eric Lander. He was until recently the science advisor of the United States president. He had to step down because of a few unfortunate things. But the main players here are Craig Venter here and Francis Collins, the chair of the National Institute of Health in America. And as you can see, a few British people are also there via the back door, and they also joined. The Zanger Center in the UK has also helped a lot, as has the Pasteur Institute, for example, in France. So it was a Europe-US collaboration. So I guess all is well that ends well, but I'll go back a bit. If you look carefully here, these two guys really hated each other because it was really a photo finish. What had happened, it looked earlier on that all would be lost. The Allied forces were too slow. Venter had all done red baker's yeast. He had sequenced in no time. He looked as being in pole position. Of course, the battle was a bit unfair because all these scientific labs, they opened up on their data, sent it round. So Craig Venter could happily tap into this, and he kept all of his stuff secret. So in that sense, it was a battle of many against one, but the one was a bit meaner or had more means to secrecy than the other party. So anyway, so it was a happy thing, and we had the human genome. Well, at that time, the first copy of the human genome had roughly 90% or a bit less was sequenced and not with great quality. So it took a few years after this to complete the human genome, and I think this was the year 2000. In 2004, I think the Human Genome Project was officially called closed. And the first complete human genome was there. Well, by then, already hospitals started sequencing and measuring DNA sequences of all kinds of patients. I draw a little bit your attention to this cake here, circle, where you see that in the green bit, a lot more than half of our genetic material, our DNA material, consists of repeats. We are big stutterers when we look genomically, and that is for a reason, a biological reason. So this is so a lot of evolution is possible because of this redundancy as a clear biological and evolutionary reason to develop new functions. But so that is what's happening, and later on this will come back. So a little bit on the two camps. So Venter against the Allied forces, the Human Genome Consortium. Venter did everything computationally. Basically, what is happening when you sequence a genome, you get very small fragments of the DNA that you don't know where they are placed. And then you need to sort of paste these fragments together in the most likely DNA molecule. I'll get back to this. So this is what Venter used, his so-called shotgun technology, where he said, I start just picking up all of these fragments and then build the complete DNA sequence. The Human Genome Consortium, they were a bit more careful. They said, well, this is too computationally demanding. So let's take big portions. We first chop up the DNA in portions of 150 kilobases, 150,000 letters. And then we do the same trick as Venter, but then we know at least that all of the snippets we measured there fall within this 150 kb. So you know better where things are. In Craig Venter, any fragment might be at the very beginning of a DNA sequence or at the very end. You need to computationally find the most likely configuration. Another problem, of course, that this leads to high error rates in how to paste these things together. And an important problem is the repeats that I just told you about that are very, very frequent, happening a lot in the human genome. Okay, so after all of this, we got next generations, literally, of sequencing, and they're called NGS, next generation sequencing. And that is all done because the process of elucidating a human genome was very slow in the first attempt for the Human Genome Project. And nowadays we can really sequence a patient or a human in a matter of 1 to 4 hours. So that has, it's so much faster than before. So, and the cost has gone down. The Human Genome Project was estimated to cost at least 10 billion American dollars. It turned out to be only cost 3 billion, which was much better than people feared. But now a human genome is at the cost of, here is 10,000 euro. At the moment, it goes down more like 1,000. And it doesn't seem to be unlikely that a human genome might cost $50 or so in the next future. And so that will make it cheaper than, for example, acquiring a passport in the Netherlands. So your DNA is who you are. So I would say everybody should just sequence him or herself and use that instead of a passport. One problem with the next generation sequencing technology that we have in general is that the fragments are a lot shorter than before. For the Human Genome Project, they were roughly 1,000 long, and NGS fragments, or and they're called reads, are more 100 long, 100 to 150 long. So the problem of pasting the snippets together in the most likely DNA sequence is a lot more complex. Here we see technology build-up, what has all happened. So it's a logarithmic scale. So, you know, one step is a factor of 10, I guess. So the read length is just, you know, how long is a snippet? The longer, of course, the easier to build back your DNA sequence. And then you see on the y-axis is the speed, also logarithmically 10 log. So you see that, you know, it is orders of magnitude. The technologies became faster. And interestingly, you see, if you can see my cursor more at the right-hand side, you see some developments like Oxford Nanopore that are now able to sequence long snippets, long pieces. And basically, you know, a few years back, you see that the first 2 million megabase DNA read was achieved. You know, for your information, a complete human DNA is roughly 3.3 billion building blocks long, so 3.3 billion letters. And so, you know, but this goes in the millions now. So perhaps in not too long, we don't need to do this pasting together problem. You know, we will just measure each of the chromosomes in one go, but that has yet to be achieved. So the problem then, we have millions to billions of sequence short fragments, and we need to paste those together to establish the most likely DNA sequence. So there are two main techniques to do that. The first one is de novo, when you have measured some genome of some animal, say, or some organisms, or the first human, then there is nothing to base yourself upon. So you have to do it de novo. Then you really have to use all of these fragments and try and build, you know, the most likely DNA sequence. And errors can be made. Sometimes you get that you can only build larger fragments and not a single fragment because pieces are missing, or the technology doesn't allow you to paste everything together. So there are complications on the way there. And that is why it's nice that, for example, for the human genome, in the meantime, there are millions or close to probably 1 billion human genomes that have been fully sequenced on the planet. And then what you can do is, if you have measured the snippets of a given patient and there is another human genome, you could ask yourself, can I now just match all these green fragments here in the picture, lower right-hand side, onto the genome of this other patient? So you use a reference genome, and then the problem is really a lot less complicated, because basically you have to align each of the snippets, and you don't have this problem of discrete portions of DNA that you're being able to build back, and so on. All of this is a lot simpler. But again, time is an issue. Scalability is rampant in bioinformatics. You know, most technologies really need to think to make it fast for the problem size we have at hand. Okay, to give you a little bit of insight, I'll gloss over it for all of the technology. So if you have these fragments, the idea is to establish the overlap. So that makes the problem, of course, quadratic, to compare all against all snippets. And then when there is overlap, you could sort of, you know, then that this one read would precede this read following it. And if you have all of these overlaps, you can use this in a problem that is known in computer science, it's the shortest superstring problem, to string up all of these reads in order to find the most likely, hopefully, the correct genome. Here is where this big problem comes about as well, that the human DNA is very repetitious, and that generates problems for this approach, shortest superstring problem, because when there are repetitions, by definition, you don't have the shortest superstring as a solution anymore. So a lot of interesting methodology has come from this problem. Okay, here you see what could happen as an illustration. So this is on the top, you see the real piece of DNA, how it should have been, with one repeat RPT1A and the second repeat, which is maybe identical or near identical to RPT1A, RPT1B. And what you see normally, if you're not careful, that, you know, of course, they are so much alike, or maybe even identical, that then all of these snippets are sort of aligned and matched to one fragment, and this region in between will be thrown out. So these are the problems you face in many cases when you have a repetitious DNA. So there are now methods and measurement techniques to alleviate these problems. We try and make longer reads that can bridge from one repeat fragment into another to do something about it. So It is working at the moment, but this is really a fundamental problem. 4, de novo sequencing, I should say, because this problem is, well, that's not a problem when you have reference-based sequencing, because you just align the snippets to a complete DNA sequence. Okay, so the question after all of this technology that became, known after the Human Genome Project, and by the way, early expectation was, when the Human Genome Project was just finished and we had the first genome, people felt that cancer and all of these types of terrible diseases would be solved within 1 or 2 years. And that didn't quite happen. The complexity of those problems are really a bit larger than that. And then people started asking, who are guys who can deal with this kind of information, with the genome sequences, and make some sense out of them. And then, of course, a few bioinformaticians stick up their fingers and said, that's us. So that made the day of bioinformatics. Funding came about, and the area has, in fact, grown tremendously from those days. And it's fair to say nowadays that if there is any field where you can be absolutely sure to get a job, well, talk to computer scientists. The job certainty is also high, but in bioinformatics or system biology, it is quite high, I might say. Okay, so why bother then? Why do this? Well, it's all about trying to find out about these all of these components in cells and how and all of the cells that sort of act together, and try and make sense how this works, be able to treat and address diseases, and with a view that cells and organisms basically are information processing systems. So one of the early problems, and I already mentioned that to you, is and this was a problem where you would get, maybe 2 Nobel Prizes or something, was the so-called protein folding problem. That is, a protein is basically a sequence of 20 different types of building block amino acids, and the average protein might be 300 amino acids or something like that. So that protein knows how to assume its 3-dimensional form. This is according to some physics, hydrophobicity versus hydrophilicity. So hydrophobic amino acids versus hydrophilic amino acids, liking water or disliking water, building a protein core away from water. There are all kinds of rules, but basically we don't understand really how the physics work, and we can't really build something together. And here you see a first big entry of computer science and AI, because there is the company AlphaFold. I think in 2014, Google started, taking part in that business. They've really used deep learning to great effect. And one of their important methods is called AlphaFold. This is just a deep learning system. They've been mining all known protein structures and protein sequences that are quite a lot, and used other and put it in a big deep learning neural network. And they're now able to predict, you give that program a protein sequence, and it builds, a likely protein structure from that. Okay. So another thing to learn is, I'll skip a few slides because I'm seeing I'm going in the, the time is progressing. We use evolution a lot. Well, I'll do this one. So common ancestry is a very important principle in bioinformatics and in life in general. It dates back to Darwin. If 2 species or 2 proteins or 2 genes seem to derive from a common ancestor, once upon a time they were the same in the past, then what you can do is the things you know for one structure, you can transfer onto the other. So there is a lot of methodology is about database searching. You try and find, you have an unknown gene, you try and find a gene in some database where people have measured some functional things about, and then you transfer all you know to the unknown sequence. And that is generally a very successful strategy. And I think 98%, if not more, of cases, this would be the correct thing to do. This is about the complexity of life. You see genes and then proteins and small metabolomes where proteins act to mostly accelerate processes as enzymes and so on. And here you see a very old picture, the Boehringer Ingelheim poster, where you see a lot of the pathways in each of the cells. I think by now this poster is roughly 10 times larger. This is one of the late 60s. It's a classic. that I'm showing you here. And so this is about complexity in cells. So we try and understand all of this functionality by a lot of networks happening in the cell. There is gene regulation, there is protein-protein interaction networks, we have signaling networks, we have metabolomic networks. I just showed you one, and there are all kinds of other networks. We need to think, certainly as computer scientists, that most of these networks are artificial. They're only happening in our mind, in our brain, and they're used to understand it. You know, a neural network or a live neural network, of course, with nerve cells is a real one, but all of the other networks really are artificial. They're only used to understand stuff. So all these pictures you see on the right-hand side are artificial networks that we use to analyze and model systems in trying to raise our understanding. Also, all of these networks are heterarchical, so they impinge in one another, and that makes understanding life complicated, but a lot of fun to think about at the same time. I'm going to skip a few things. So a lot of technology here is multiple sequence alignment. So comparing sequences, I already mentioned that, is really very informative because you can get a feel of which parts of the sequence are conserved and which parts are more variable. You see, for example, on the bottom that there is a red element. These are all kinds of different sequences from different organisms. And you see that this fragment of these sequences is totally conserved. And that is indicative for the importance of this fragment. So probably this little fragment here is important for the structure or the function of this protein, for example. This is a protein, by the way. You see 20 different letters as amino acids. Okay, and then there is a relationship between such an alignment and a three-dimensional structure of a protein, because this matched letter here might well be a letter that happens somewhere at a defined position inside this protein. So alignment will map onto pieces that are outside of a protein or inside of a protein. And to do this exercise and relate it to things, we can really learn a lot about the relationship between the sequence and the structure, and from that, the function of a protein, for example. Also at the network level, we see a lot of evolution. Here you see, for example, a comparison between human and yeast, and you see that a highway has been created in yeast as compared to the human. And so you see that also networks have evolutionary origins, have evolutionary motifs, but a lot is happening. And so network alignment is also a field of study, and trying to compare one organism with another one at this level is also very important to find out about the function and how things really work. I've skipped this example. I only show this is C. elegans. I'll show you what we can do in modeling a little bit. Just this picture, I guess, for now. So this is a multicellular system with 6 cells, and you see a whole machine in each of the cells. So we are trying for smaller systems to build everything that we know together and model this through times. Incidentally, these models were done using Petri nets, and that is something that is used widely in many fields of computer science. Okay, so I'm trying to draw a little bit to a close. I'll do a bit on data and a bit on the importance of data. So, as you all know, databases grow exponentially. You see that here illustrated. Welcome to the Yotta world. The data is the new oil. So data management and all of this is becoming really all important also in bioinformatics. And but one thing is with data, it can grow. It grows exponentially, as we know, but it can also evaporate. Here we see an old picture in 2013 in Nature, where they looked at the supplementary information at papers, and how you know, at those times you could store them in on your own systems, and it turned out that the vast majority of these data are gone before you know it. So data management is a big task in the whole of science to do that well and store your data and be able to use it. So many times you say, oh, there was a link, laptop of a former PhD student, and I don't know where the person is right now. So that's not a great situation. At the same time, data grows exponentially. And for example, DNA sequencing data is growing much faster than the Moore's law rate of doubling every 18 months. The doubling rate of these sort of data is every 5-6 months. and now it tends to go to every 4 months, the data doubles. So it's clear that we have a formidable task here to store and to manage all of this data. Okay, a quick piece of information, by far the best disk or entity or thing to store data is DNA, because you can store 1 yottabyte of data on roughly 1 gram. One of the champions of doing this, storing information in DNA, is George Church at Harvard, and he has managed to store a PDF file of one of his books in a DNA sequence, and it could be sequenced and read back and be printed. So that was a big success. In general, the problem, of course, is we could start doing this. We could store. We never had a data problem anymore. We can store roughly everything in DNA, much better than disks, computer disks. But the reading out of the information is getting better by the day. But writing DNA, we need to build a DNA molecule, is still problematic. So I think we'll stick to computers for the time being. So a little bit on data and the importance of that. So we live in an information era with data science, and this has fundamental, a profound influence on society and on science and health and the quality of life and so on. Bio-based economy, for example, cannot be done without data science. And a lot will come from this. We're predicting the spread of disease. We can do flu. COVID is a bit more difficult at the moment, but we can do weather prediction. Is getting better all the time. In physics, all kinds of developments are being taken. A few years ago, we discovered really the Higgs boson was confirmed. And in social trends, this is sort of an early data science success in 2015 on the bottom of this slide, where it was decided that Jay-Z would move his concert from Stockholm to Gothenburg because there was a much bigger fan base there, and that was a great success, according to some of data science. Okay, what's the importance of data? I always like this tagline. Too often we forget that genius depends upon data within its reach, so that even Archimedes, although he probably was a clever man, could not have devised what Edison did. And data is all important. So that's why I would like to draw your attention to the FAIR data principles, and I'll gloss over that, mostly where data should be findable, accessible, interoperable, and reusable. And this has to do with persistent identifiers, with accessibility formats, and so on. And but maybe most importantly, for science to drive further is the tagline, to go further is the tagline, using other people's data for a new purpose. So reusing data for a new purpose. And that is important for complex science. And, I think the solution should be developed within computer science of all this. So semantic web solutions. And so this is a field of study also in bioinformatics. You're talking about community supported ontologies, and all this is to make the data interoperable and reusable. FAIR data has to do with storing and longevity of data storage and so on. Here you see a bit of the history. FAIR was coined for the first time in the Netherlands, in Leiden, and the whole world in the meantime talks about this. G7, G20, all kinds of initiatives, funders, science funders that embrace the principle and so on. So, there is a bit of stick from funders to start thinking about all this and become active at this. But also there are lots of bottom-up initiatives that are more important scientifically. Okay, I'm going to skip all of this. So from this, the idea is if there is FAIR data, we have the basic stores, higher levels of FAIR data, then the networks we can use, and all kinds of analytics that will be enabled by FAIR data. And the idea is, so basically the data should be distributed and be reachable through FAIR data points. And thanks to a lot of developments, I'll come back to that for COVID, that this is now becoming a reality. What you can do is build knowledge graphs. Most people in computer science have heard of this, where you can start reasoning across knowledge domains and find important overlaps between, for example, malaria bug and malaria as a disease and the bug. And the one knowledge domain might be about malaria and all there is to entertain there, and the bug, and then you see that you get overlap and knowledge that could be made into actionable knowledge. So, COVID was, of course, a point in case here, because the last 2 years, many patients abounded on COVID, and the need for fair data was larger than ever. So a lot of quick developments happened. For example, in the Netherlands, we used the case report form according to WHO standards to be able to quickly report what happened with patients, how ill they were, what was happening, and make that this data could be pooled so that we can learn much quicker than we did for COVID for a next time when there is a bad variant coming up, or new virus types. The Marburg virus is one of the things that could start happening, or the Nipah virus. Look them up, don't get scared. But these are next viruses in the list of things that are going to happen. So to be prepared for it, to have an information structure and a data structure, infrastructure in place to be able to build up knowledge quickly will be vital for human existence. Okay, last little point, personalized medicine. What can we do with all of these genomes that we have of patients? The idea is that we compare like for like. There are 4 possibilities when we prescribe a drug. The best would, of course, be that, the lower right corner, it's not toxic and it's beneficial, and you have the other options that are less beneficial to a patient. So how do we get the knowledge of what works at a personal level? And the idea is not to wait until disease starts, but to have markers, measure it, and be quicker than the disease, and remediate, have a healthy lifestyle, have a good diet that is there helping you as a person to not get some diseases that have that impede the quality of life. So I guess by this I'll go to the last slide. What I want to say, maybe some of you know about Peter Kovner. So he said, in modern sciences, you cannot just be an expert in just one area anymore. I'm putting it to you, you can agree or disagree, but at least in this, that is true. Old-fashioned chemistry cooking is over. We need to think broader and wider, and in order to tackle the big scientific questions we're facing today. And with this, I maybe have to apologize for being a bit over time, but I would like to leave it here, and I'll be happy to take any questions you might have.
Okay, thank you for this very energetic and comprehensive overview. Let me see where our people. If you have questions, then of course we can first add our either virtual or our real applause. So thank you very much. And if you have questions, please raise your hand, and I will go through these in the order that the screen that I have. I have an initial question, and that is, you said that being schooled in bioinformatics means guaranteed employment.
Yeah.
So what do these employed bioinformatici actually do?
Very good question, thank you. I've tried to sketch sort of the type of question. So you can, you have sort of technicians in bioinformatics. People are really very good at the computational side, even the compute side, cloud and so on, all the way till far more applied bioinformaticians who know a lot about certain diseases or about cells. Very often, these bioinformaticians are embedded, for example, in hospitals. So really, it's manifold. There are all kinds of environments where you can find employment, and also sort of the focus of what you do is also has quite a large variance.
But so trying to make a distinction between other data-driven sciences. If you're studying environmental sciences, where you're looking at processes of pollution control, not so much disease control, but there's also a big data policy. If someone's interested in studying bioinformatics, what kind of correlation is there? What tools do you need to bring in? Are you looking for biologists who you retrain to be computer scientists, or are you looking for computer scientists who you're going to retrain to be biologists?
You really look at both. So, machine learning skills are, of course, very, very important. Software development skills are very important, but also knowledge about the domain of study. So basically, a biologist that gets retrained into computer science is still another person in many cases than, computer science, learning more about biology. And basically, you need both of these types of people here. So when you compare it to environmental science, for example, also that, of course, is a very broad area. But, the complexity and the difference of scales we find maybe, is really formidable. And what we find, for example, in our master program, we do have a master program in bioinformatics at the VU, that we talk a lot about transferable skills. A lot of the people that come out of a program find jobs in insurance companies and even banks for that matter, because, their machine learning skills and their insight in complex processes could also be, applied to stock market principles, right? So we see that as well. So, and the same holds
making people better, and the other is making people worse. Right.
Sorry?
One is making people better, and the other one is making people worse. Right. Personalized.
Yeah, maybe. Yeah.
Right. Portfolios.
Okay.
And could you say something about the future of personalized medicine? If you look forward a decade or a century, what's the future of applying techniques to my genome? I probably won't be here in a century, but I hope I'll be here in 10 years. So that, are these Star Trek things where a scanner comes in my belly and then tells me what I had, what I have, and what I'm going to get?
Yeah, perhaps, you have just a little card, you don't have a passport anymore, your genome is sequenced, you have a headache, you go to the GP, he measures a few things and says, oh, I see what's wrong, it's this here, this small molecule, your metabolome is a bit disturbed there, we know an exact, quite a drug for this, no problem, no side effects, it really cures, and maybe later in the evening the drug will be delivered on your doorstep and you can just do this. So that sounds great. So, but that would mean we get N=1 medication, that the drug should be manufactured for your personal problem. So if you go with this to big pharma, they say, we're not interested in this. We are interested in generating aspirins, low R&D cost, and everybody takes it. That's a good business model. N=1 medicine is not something. So a way to address that is combination therapy, and there are more possibilities. And that is also a reason from the negative, but anyway, that people are looking into food and nutrition and lifestyle, because this, the repertoire of personalization of what you can do is increased by that. And also, incidentally, it also is about being earlier than a disease really happening, avoiding all of these type of things. So I guess in 10 years' time, it will all be a bit different. And I think genomics will have gone into the clinic. It's already starting for cancer, immunotherapy, for example. We'll see a lot more of this. And I hope that a lot of the diseases that are now lethal will be chronic, if not better.
Okay, and then perhaps the last question, if I look at the clock. What's the major pushback on bioinformatics? What are groups going to be demonstrating against outside of City Hall? Is it the privacy of personal sequencing? Is it the development of control drugs? Is it that it's using up all of the energy that's left on the planet? What is it that the people are going to be complaining about?
Yeah, of course, personalization. Data privacy issues are there. That's more, a problem in medicine. So we need to think about changing the culture. For example, that the citizen, a patient, has control over his or her data, and so on, and issues like that. What we have experienced already, maybe at the beginning, just after the Human Genome Project, we were promising too much. We were happy as bioinformaticians, now we're in the limelight, so let's promise a lot and we get a lot of money. And, you see that, and then there is disappointment. You get the valley, right? And so then we got systemology, people claiming, oh, we model the things, we'll understand processes through time. Bioinformaticians label things more or less. They say this protein has this function, but that is not the same as understanding processes through time, how they develop. So that was the realm of systems biology, and now we see that they shifted us away a little bit. They took a lot of funding, and now so all the time you have hypes and fashions, and that is a threat maybe to science in general. When everything is too important, you get politics in the equation, and so that's so I would say that. I would say technology developments, we don't know really what is going to happen, and the consistency of developments. The data is so diverse that finding consistent models, being able to really predict and do something for a personal citizen is still quite a challenge. A lot of people talk about digital twinning and so on, which comes out aviation industry for patients now. So you have a perfect model of yourself, and then anything can be tested before things happen for real. So I guess the problems there and things that could go wrong perhaps might be problematic in the future. But if we're careful, that won't happen.
Okay, we've got a question from Alina that says, what aspects do you think are important for decision-making recommendation systems in drug use for treatments?
That's a very, very good question. I would say this is a fundamental question, of course. So who's to blame when a treatment doesn't work, right? Is that the deep learning system, or is it a doctor? So there is a lot of this. I think that what is quite important, certainly for the time being, is that these that tools, technology, and human knowledge is used in tandem, is used together. That is coined now as a field of hybrid intelligence, where technology just is working seamlessly together with the human brain. So when it comes to decisions, I think digital twinning will be important, where you can try out more different medicines. When you do it in the real, there is always an element of risk there, that something might be toxic or not work, or even worse. So you can test that. So I think there is really a lot to be gained here. And I think that the role of machine learning and decision systems will be all the more important. It's already quite very important, but that role will grow in the future, no doubt.
Okay.
I hope that answers a little bit the question.
One of the decisions that I have to make and that I'm responsible for, and which I will take all of the blame, is that we're out of time.
All right.
I would like to thank all of you who have started with us and ended with us, and especially Professor Heringa. Jaap, thank you for this dazzling overview. There are a lot of sub-threads and a lot of sub-threads that we can sequence in our own minds on this. But this gives a nice impression of what it is that people in the bioinformatics hallway are doing within computer science departments. Thank you very much for everybody who's watching. We hope to see you next month. There'll be an announcement beginning of next week on the next set of lectures that we'll be providing in this distinguished lecture series. I thank you all, and I wish you all a good weekend. Thanks.
Thank you. Bye.